All right, welcome to the second of our Honors Biology podcasts. This one is about viruses. This is beginning our survey of living things, which is just kind of taking an overview of the different kinds of living things on Earth. And we start that journey with viruses, which is kind of interesting because really, by most folks, they're not considered living truly. At least they don't meet the definition that we set up in the beginning of the year. We said that that a living thing had to be made of cells. It had to be able to grow. It had to be able to use energy. Had to be able to reproduce and so on. And viruses cannot meet all of those criteria. So technically they're not living. Does that definition really matter? I don't know. They sure do cause a lot of trouble in living things, that's for sure. So let's talk about viruses to get us going. They are extremely tiny, 1/100th the size of the smallest bacterial cell. So we are talking about sorry, I've got to get my pen working. Very small things. This diagram is showing you one of our cells, an animal type cell, and it's comparatively enormous. Remember, this is still a single cell. A bacterium is much smaller than that. Viruses are much smaller still. So very, very tiny, tiny things. Technically, as I was just talking about, they are not alive. They cannot grow. A virus is one size, and it's always that size. It can't do cell respiration. It can't do photosynthesis. It can't do any of those kinds of complex chemical processes. Can't do any of them. No metabolic processes. No chemical reactions in them. Now, the only thing that it can do that suggests life is it can reproduce. But even that, it cannot do outside of a host. It requires a host cell. So a virus is very much a parasite on other things. It requires a host cell in order to reproduce itself. Okay? Like one of your cells. Naming conventions, they're not named like living things are named, going through all those kingdom, phylum, class things and the binomial nomenclature. They don't do that because, again, they're not living, so they're not classified or named in the same way. Latin's not used. They are named for the disease that they cause or for the tissue in which they cause infection. For example, rabies virus is named for the disease rabies. Polio virus is named for the disease polio that the virus causes. Adenoviruses cause a lot of different ailments in folks and other things as well. They were first discovered in the adenoids, which is a glandular tissue, I believe, inside your body, but they infect other things beside that, but that's what they were named for. Bacteriophages, phage is another word for a virus, and bacteriophages are viruses that infect bacteria. So even the lowly bacterium can get infected by a virus. So can plants. Anything can get infected by a virus. Okay? Now, you really do need to understand the structure of a virus. It's important in how it functions. It is made up of a protective coat made of protein, and that's what I'm drawing here. This coat made of protein, which you see drawn better down there, this is called a capsid, and it is a protein protective layer. Inside the capsid, you find a molecule of DNA, genetic material, just like what you'd find in your cells or any other cell. Interestingly, in viruses, the genetic material can just be RNA. Okay, and we'll talk about why that's important later on. So you need to know that it's got some genetic material, could be DNA, could be RNA on the inside, the protective protein capsid on the outside. The shape of the capsid is important. We'll talk about that in a minute. Some viruses also have a third layer around them called an envelope. We won't talk about that, but human flu virus, for example, has an envelope around it made out of actually cell membrane from the cells it infects. So, these are just electron micrographs showing you all the many different viruses that are many, many more than these. This one is the one that we just drew a second ago. This one is a bacteriophage called a T4 bacteriophage. It infects bacteria. And this is an electron micrograph showing you, it really does look like this little alien spaceship. It's kind of creepy. This is a human flu virus right here that makes us sick in the winter usually. This is an adenovirus we were just mentioning. This is a virus that infects plants called tobacco mosaic virus. But again, anything can get a virus. There are viruses that attack all kinds of living things. Now, the shape of a virus, in particular its capsid, is really important to its ability to infect a cell. Remember, that won't do. Remember that our cells have certain receptors on them that may allow our cells to engage with other molecules in their environment. Maybe they recognize something that's foreign and avoid it or attack it or whatever. So there are receptors on cell surfaces, okay? Maybe these receptors allow for us to bind molecules we need and take them into our cells, whatever. They do all kinds of things. But the protein coat or capsid of a virus is designed to fit. Maybe it fits right into, if that's the virus, maybe it fits that receptor. That's not because we want the virus to fit our receptor, but because the virus has evolved to fit a particular receptor. Why? Because it allows it to gain entry into that cell and take over that cell and use it for its own purposes. Now, what this means is that viruses are very specific to a cell type. For example, rabies virus only infects nerve tissue in mammals. That's it, because that's the only tissue that has the receptors that the virus recognizes. Now, if a virus mutates, then it might be able to infect some different type of cell, but that's another story. Okay? All right, we're going to take a moment and try to understand how a virus infects your cells, and we're going to look at something called the lytic cycle. Now, you should have two diagrams. They look sort of like this, but they're not. They're hand-drawn by me. So what I'm going to do is try to use this to help you fill out those diagrams. If you can't figure out this, we can do it in class. It's not a big deal. But let me get my... All right, so the steps of a virus infecting your cell, okay? This is actually looking at a bacterial cell because this is that T4 bacteriophage virus, but it would be essentially similar. All right, this brown molecule here, that is the DNA of your cell. This is the virus, of course. And the first step of a viral infection is that the virus attaches to the cell, okay? And it does that because it matches the specific receptors on the cell surface. Now, the second thing that happens is, as you can see, the DNA that is contained inside the virus capsid is getting pushed into the host cell. So, the virus injects its DNA into the host. Okay? Now, once that DNA is in there, it does something. Okay? It basically destroys the host cell's own DNA, which you can see here. This is the host DNA. Okay? And it's getting all chopped up. All right, and the capsid is basically just an empty vessel left on the outside. Now, what happens at this point is that the virus DNA is the only DNA left to tell the cell how to function. The host cell DNA is gone. That's done. So now the viral DNA is going to be instructing this cell in how to function. And what does it do? It tells the cell to make viruses. It tells the cell to use its cellular machinery to make virus parts. So it makes the DNA, it makes the capsid, it makes all the landing gear here, it does everything. And so we end up with a bacteria or a cell full of virus parts. So here, let me back up a minute. So in this diagram, the host DNA is destroyed by the viral DNA. Here, the viral DNA tells the cell to make more viruses. And so they make the virus parts, the viruses assemble, and eventually we make so many virus parts, we just keep making them, because that's what the viral DNA is telling the cell to do, and eventually, guess what's going to happen? That cell is going to go, it's going to just pop and release all those viruses into the environment. And then guess what? There's those viruses that can infect more cells, and thus the infection spreads. While this is showing you a bacterial cell, infection in you happens very much in the same way. So here the cell bursts, releasing more viruses. Okay? And then we just start it all over again. So that's basically how a viral infection works. If this doesn't make sense with the diagram you have, don't worry about it. I'd be happy to tidy this up in class for you. Okay? All right. So let me go back just a sec because I want to make sure we've got our steps. This is called the lytic cycle of viral infection, what we just did in that diagram, because to lyse, and I'll show you the word lysis, or the verb is to lyse. This means to rupture, to burst, to make the cell burst, and that's how this cycle ends. So these are all the steps. The virus lands on and attaches to the cell. It injects the nucleic acid, the DNA, the genetic material. The host cell DNA is destroyed. The viral genes hijack the cell and force it to make viral DNA and capsids. The viral components assemble themselves into new virus particles. And then the cell undergoes lysis. It bursts, and the viruses come out. The virus particles then get to infect more cells, and in 90 minutes, you can end up with 200 new viruses. And those 200 new viruses can infect 200 cells, and then guess what? 90 more minutes, each of those 200 cells is releasing 200 viruses. So you can see why the infection gets very serious very quickly. Okay. Now, there is a second cycle, however, called a lysogenic cycle. And in this case, the virus does infect the cell, but it doesn't immediately kill it. Okay? I don't like this drawing. Let me see if I can draw something better. Let me get this blank slide. Okay. All right. So essentially in the lysogenic cycle, what you would have is it would start out just like the lytic cycle. The virus would attach, and then it would, I'm drawing really sloppy, sorry. It would inject its DNA into the host. Oh, remember the host has its own DNA particles in here. So here's the host DNA. So then here's where it differs. Let me get myself a slightly different color of ink here. This can be the DNA of the virus. So that's virus DNA. Now, instead of chopping up the host DNA, what this virus DNA does is it embeds into the host DNA. It just becomes a part of it and kind of disappears. The bacteria, the animal cell or whatever cell it is does not recognize it's there. It just stays there. That means as this bacterium is living out its life, doing its thing, as it's reproducing and making baby bacteria, okay, as this cell is going to do that, it's going to make more cells as it goes through its life. Every time it makes more cells, it's going to make more cells that contain that virus DNA. So that virus DNA is going to show up in each of these, and so on and so on. So every time the cell reproduces, not only is it reproducing the cell, but it's reproducing the virus. Well, eventually, we don't really always know why. Some people say it's due to stress. Environmental stressors can cause this to happen, but we're not sure. But something will cause this virus to come out of being dormant. It will stop hiding. It will come out of the host DNA, it will come out of the host DNA, and then boom, we start back with the lytic cycle. It does all the same things that the lytic cycle did, ending with the cells, now multiple cells, because many cells have got this virus in them now. It will end up with a bursting situation. So the key difference between this and the lytic cycle, by the way, remember this is called the lysogenic cycle. The key difference is there is a period of dormancy where the virus just hides away in the host cell. Okay? So let me go back here. All right, so the steps of a lysogenic cycle infection, again, virus attaches, injects its DNA, but here's the difference right here. The viral DNA becomes part of the host's own DNA. And every time that host cell reproduces, it makes new cells that have that viral DNA in them. When that is the case, that DNA is called the provirus. It's not active, it's called the provirus. And this can go on for a while, but the virus can pop out of dormancy at any time, and that lytic cell can go into effect. Some viruses that behave like this are cold sores, herpes simplex 1. If you ever have had a cold sore, yes, you have had herpes. And guess what? You still have it because it remains dormant in those cells even after the cold sore has gone away. What causes the cold sore to become active again? What causes it to enter the lytic cycle instead of being dormant in that lysogenic cycle? Who knows? Some people say stress. Who knows? Chickenpox is an example of a lysogenic cycle virus. If you have ever had chickenpox, you still have it. But it is dormant in your cells. It is hiding in nerve cells. And if it is reactivated, it comes a particularly nasty illness called shingles in adults. And so we can talk about that more in class. HIV, the virus that causes AIDS, is a lysogenic virus. It can be hidden in white blood cells inside their DNA for an indefinite period of time. But when it goes into the lytic cycle, then the patient starts to show signs of AIDS. It starts to kill those white blood cells, and then the person cannot fight off infections. Okay? All right. Some viruses can have RNA instead of DNA as the genetic material, and they operate a little bit differently. Basically, if this is the case, the RNA would enter into the host cell. So the RNA would enter the host cell, but it can't do its job as RNA. It has to become DNA so that it can tell the cell what to do. So it comes packaged with an enzyme. It's called reverse transcriptase. Well, ACE tells you it's an enzyme. It's going to reverse transcription. If you can remember when we were doing our genetics unit, transcription is when we go from DNA to RNA. Well, guess what reverse transcription is? It's going from RNA to DNA. So, in this case, the RNA from the virus gets changed by reverse transcriptase into DNA, and then it can either tell the cell what to do, or it can go dormant inside the DNA of the host cell and hide for a while. Okay, so makes DNA from RNA, inserts it into the host chromosome, and then we get the lysogenic cycle. Usually that's how it works. Okay, so that's how an RNA virus can work. And interestingly, there are lots of RNA viruses that are very important to us. One of them is the flu virus. RNA viruses tend to be really unstable because RNA is not as good a molecule as DNA as far as stability goes. Remember that DNA is this double helix, and so those nucleotides, the bases are really well protected by facing into each other, but RNA is just a single strand with those bases just facing out into the environment, and it's much easier to mutate this, so RNA viruses get mutated and change very easily. Flu virus, this is why we have to have a new flu shot, flu vaccine every year, because flu viruses mutate like crazy, and they're RNA viruses. HIV mutates like crazy. That's why it's so hard to get it under control, because people will take medications, but then the virus mutates and becomes resistant to those medications. So RNA viruses are highly variable, easily mutated as well. Where did viruses come from in the first place? Were they the first organisms on Earth? Answer is no. In fact, what scientists think is viruses actually came from the cells that they infect. Viruses seem to be more closely related to the cells they infect, meaning the rabies virus seems to have a relationship to mammalian nerve cells, for example. It is thought that viruses originated at some point in history from the host's own cells. Perhaps nucleic acids, DNA, RNA, escaped from the host cell, whatever, got into the environment, and this is the way they've found to reproduce and stay part of the living, quote, experience. They have developed a way to reproduce as parasites of that same host. Obviously, this was not thought out or intentional. It's just that this is what DNA does. It copies itself, it reproduces, and if a way happens for it to do so, this is what it does. All right, so how do we get rid of viruses? It's really hard. Once you've got a virus, it's really hard to get rid of it. And generally speaking, if you go to the doctor and you've got a virus, he's going to basically say you're just going to have to let it run its course. There's no way to really kill it. They're trying to come up with ways to do that, but we don't have very many good ones yet. The best weapon we have against viruses is vaccines, meaning we get a shot, an injection of a vaccine that makes our bodies resistant to that virus. Okay? What is a vaccine? A vaccine is a dose of medicine that is basically made of killed virus or damaged virus that cannot cause disease. Okay? We inject it into our bloodstream, and what that does is basically it's like you're injecting the capsid, for example, into your bloodstream. It doesn't have any usable DNA in it, but guess what? Your immune cells, your white blood cells, here, I'm going to draw a white blood cell. Here he is. He's like, I'm going to eat you. So this is a white blood cell, and it is going to learn what that virus particle looks like, and that way, if you ever see it again, like you really get infected by this virus, he is already going to know what it looks like, and he's going to be able to attack it quickly, and you will not get sick. Part of the reason you get sick is because your immune system, your white blood cells, have to learn what the infecting agent is, and then they can fight it. Well, this way, by injecting a vaccine like that, you're teaching your immune system what it looks like, and then it will be ready to fight it if you're ever exposed to it again. Okay? So you're kind of getting your immune system used to seeing it without ever having to get sick, which is a good thing. So the immune cells learn to recognize and fight any invader that looks like that virus, and you don't get sick from exposure to the virus. Vaccines were invented by a guy named Edward Jenner. He developed Edward Jenner. He developed the first vaccine, and it was to fight smallpox, which was a deadly, deadly disease. It would cause horrible scarring in people. It would very often kill them, but if it didn't, it would leave them horribly scarred. A very deadly, nasty, nasty disease. But what Edward Jenner noticed was that milkmaids, women that worked with dairy cows, milking them and so on, did not ever seem to get smallpox. But what they did get was something called cowpox. It makes people sick, but it is not deadly. It's like you get sick, you get better, you're okay. Okay? So what he thought was that somehow cowpox was making these milkmaids immune to smallpox. So to try it, he went and found milkmaids that had cowpox, and they would have these blisters on their skin, and he got some of the pus from those blisters. And he found a boy, and the old fable was that it was his own son, blah, blah, blah. Not quite. It was actually a kid from an orphanage, so do with that information what you will. But he gave the cowpox virus to this kid, and the kid got cowpox, and he got sick, but he got well, okay? Then what he did was he exposed the boy to smallpox. And lucky for the boy, Edward Jenner was right. The boy did not get smallpox. The exposure to cowpox, apparently the cowpox virus is similar enough in the way it looks that the body, once it learned what cowpox looked like, it could also recognize smallpox and would fight it right away, and the person didn't have to get sick with smallpox. So if you got exposed to cowpox, your body was then immune because your body knew what that cowpox and the smallpox virus looked like from that point forward and could fight them. This is a picture depicting Jenner doing his experiment with the young boy. So we already talked about this. Why did it work? Cowpox was similar enough to smallpox that the immune cells were able to recognize and fight both cowpox and smallpox viruses after they'd been exposed to cowpox. Okay? So, when you get a vaccine, generally speaking, lifelong immunity, sometimes they wear off, and we need to look into why that might be. But as long as the virus is stable and does not mutate, and that's key. If the virus mutates at all, then the immunity you got with that vaccine isn't going to last, because if the virus changes, then your body doesn't recognize what it looks like anymore. Okay? This gets to our discussion before. DNA viruses are far more stable than RNA viruses. This means that vaccines against DNA viruses work really, really well and for long, long periods of time. Vaccines against RNA viruses work well for a short period of time until the RNA virus mutates. Okay? Smallpox has been eradicated from the planet because they got vaccine out to everybody in the 60s. People went into the far reaches of Africa, everywhere this virus existed, and gave everyone they could find this vaccine against smallpox. And because the smallpox virus is so stable, it is a DNA virus, they were able to eradicate it. It no longer exists except for in some vials tucked away in the United States and in Russia. RNA viruses, however, are not so much. HIV, flu mutate often. Hard to develop vaccines for them because they mutate so often. You got to take a new flu shot every year because that virus mutates. All right, that's it. We will talk about this more in class. Have a great day. See you next time.